see clear zones around this corner. They are named hemolysis zone. Cyclophosphamide activity. Biochemical properties. The biochemical activity of staphylococcus is high. They produce saccharolytic and autolytic enzymes. Means they are able to ferment many sugars and they are able to make protein. Fermentation of mannitol and glucose under the condition is for diagnostic value. We should detect many enzymes, saccharolytic enzymes on his mirror or by another in aerobic conditions, but do remember they are facultative anaerobic. So they are able to ferment sugars in aerobic conditions. The fermentation of two sugars is important for identification of staphylococcus. Antigenic structure. The following antigens are of practical importance. All antigens substances peptidoglycan teichoic acid, the capsule antigen, often it is polysaccharide microcapsule, and the most important are exotoxins. Those are produced by Staphylococcus. Exotoxins are protein, and they differ from antigens. The specific antibodies are produced for exotoxins. Those are named antitoxins. Viral receptors, adhesion factors, they are able to attach to the human by using of cell wall components like teichoic acid proteins and capsule, and antiphagocytic factors such as capsule proteases, protein A. By using of protein A, they are able to bind antibodies by wrong part Fc fragment. They are able to produce proteases. Then those are able to destroy immunoglobulins, cell parts, and these enzymes are able to destroy them. They have protection factors. They are able to protect themselves. Negative this enzyme, plasma carbonase, very important enzyme, which is named carbonase. But really, we are talking about plasma carbonase. Plasma is liquid part of blood, and staphylococcus have no macrocapsule, but they are able, exactly, Staphylococcus aureus species bacteria are able to produce this enzyme. Other two species are carbonate negative, and Staphylococcus aureus species bacteria are able to produce this enzyme carbonase. And due to this enzyme, plasma becomes solid around every cell, is coagulated. And every cell is surrounded by a false capsule. Capsule is not bacterial capsule, it is capsule that is developed of coagulated plasma, so they are protected from the immune cells and they are protected from antibodies. So very important enzyme in diagnosis of staphylococcal infections. This enzyme should be detected. The lecitinase enzyme that is able to destroy lecithin. And lecithin is in human being cells. So by using all this enzyme, staphylococcus are able to destroy cells. And by using all the cells and egg yolk, it is possible to detect this lecitinase because of in the yolk the same lecithin is so salt sugar is used for detection of for conservation and detection of lecitinase. Beta-lactamase, name is able to produce beta-lactamase enzyme that is able to destroy beta-lactam antibiotics such as penicillins, cephalosporins. Staphylococcus and gram-positive bacteria, they should be sensitive to such antibiotics because of the mechanism of action of beta-lactam is a disruption, suppression, inhibition of production of peptidoglycan. But if bacteria are able to produce beta-lactamase, they are able to destroy antibiotics, so they develop resistance. Staphylokinase, fibrinolysin enzyme that is able to destroy fibrin as and by using all these enzymes, staphylococcus are able to spread inside the deeply in tissues catalase hyaluronidase destruction hyaluronic acid and genase. Staphylococcus, by presence of the plasma coagulase enzyme, are divided into coagulase positive Staphylococcus aureus bacteria and coagulase negative. Other two species are coagulase negative bacteria. Aggression factor, exotoxins. Staphylococcus are able to produce different exotoxins, toxic proteins that have different action, and clinical symptoms are developed due to the 13 exotoxin. Hemolyzins are able to destroy red blood cells. Leukocidin is able to destroy leukocytes, and other cells are destroyed. Daily such toxins and toxins now are named membrane toxins. Also, they are able to destroy many cells. And exfoliatants A and B, they are able to destroy just upper layer of the skin, and bubbles are here on the skin, like due to flame action, for example. Infant syndrome, for example, the toxic shock syndrome, that is able to develop shock and patient is able to die immediately. And some strains are able to produce enterotoxins. Enterotoxins act on digestive tract, and due to this enterotoxin action, vomiting and diarrhea are developed. Such strains are able to cause food poisoning. And unfortunately, this enterotoxin is heat stable, very unusual. It is a protein. All proteins are heat label, of course. The temperature 58 degrees by Celsius. By using all this temperature, it is possible to destroy proteins, but not this one. It's possible to destroy this enterotoxin produced by Staphylococcus even by using of autoclave, even by boiling a few hours. It's very unusual resistance. Staphylococcus are quite resistant to drying, heating, and insulation. Sensitive to the action of most antiseptics and disinfectants. Epidemiology. Staphylococcus are representatives of normal human and animal microflora. They are opportunistic microorganisms. In the human body, they live on the skin. Many other species of Staphylococcus, and including Staphylococcus like Staphylococcus epidermis, sometimes they are detected on the skin, not only for the parasite. The source of Staphylococcus infection is a person, sick or carrier. Less often animals, for example, cows with mastitis, if it is caused by Staphylococcus, Staphylococcus can be transmitted by the drinking of the raw milk, which leads to the food poisoning. If milk is boiled, ways of transmission. Staphylococcus are transmitted by air, more often by airborne droplets, and by contact through the hands of personnel, through non-sterile medical instruments and wounds. Also by ingestion with dairy products and confectionery, if they are prepared or meat infected. Clinical manifestations. Staphylococcus do not have organ transmission. They have no organ stability. They are able to multiply in any organs of human body in any tissues. And they are able to cause more than 200 clinical forms. Virulent inflammatory processes of various location, causation, and severity from not limited to severe generalized, such as sepsis and septicopenia. I'm going to remind you, sepsis, of bacteria in blood, is very severe clinical. Sometimes not possible to save the life of patient. Septicopenia is multiplication of bacteria in blood and development of inflammation response in internal organs. The most severe clinical form. And they are able to cause food intoxication. Staphylococcus are in hospitals or able to cause pathogenic healthcare associated. So they are able to cause pneumonia, mastitis, bronchitis, sore throat, sinusitis, meningitis, endocarditis, arthritis, osteomyelitis. So any parts of human body are involved in these diseases. Also, they cause tuberculosis of the skin, saturation of bronchitis, media, and so on. Staphylococcus cause the following syndromes: mild infant syndrome, mild skin syndrome, and toxic shock syndrome. Immunity. As a result of transferred infection, unstable immunity develops. Microbiological diagnosis. The material conferred depends on clinical form. Baby fast, mucus from nasopharynx, baby blood in case of sepsis or septicemia, food intoxication, cold and vomiting. The rest of it should be. Diagnostic methods. Microscopy. The first material used to make smear and stain according to the brand. That's what we are looking for protein clusters and cultivation. The specimen is inoculated on yolk salt agar for detection of lecithinase or GC. If we see some unclear zones around the colonies, it means that these bacteria are able to produce lecithinase. And due to lecithin destruction, the medium becomes unclear. On whatever we will see hemolysis zones, clear zones due to red blood cell destruction. But not all strains of Staphylococcus aureus are able to produce lecithinase. The only 75% of strains lecithinase activity, so are the 25% of strains of Staphylococcus aureus are not able to produce lecithinase. So if there are no lecithinase activity, it doesn't mean that there are no Staphylococcus aureus colonies. And the most important thing is plasmacarbylase detection. Identification is carried out by presence of lecithinase on yolk salt agar and plasmacarbylase after glucose installation. And the fermentation of sugars in aerobic conditions and fermentation of many-tolerant glucose in anaerobic. Detection of plasmacarbylase plasma is liquid part of the blood, and it is produced. It is commercial, it's industrial plasma of rabbits, rabbit blood. And we have to either after glucose installation, we should inoculate bacteria in test tube with plasma, and this test tube should be incubated at thermostat, and every 2 hours we should check. Is it leaking or not? If in 2 hours, then again in 2 hours, so 2 hours, in 4 hours, in 6 hours, in 8 hours, we should see. If it's still leaking, means plasmacarbylase is not detected. If it doesn't leak, now it is solid, which means positive result, plasmacarbylase is detected. When Staphylococcus aureus is isolated, H typing is performed by use of typical Staphylococcus bacteriophages. Staphylococcus bacteriophages for typing, and exactly just for in case of isolation of Staphylococcus aureus, not in case of isolation in other species. And we have to know what by numbers or bacteriophages, what page type was isolated, and it is necessary to identify the source of infection. It is possible to find who is the carrier of the most virulent strain in case of outbreak at hospital. Then, of course, we have to do an antibiotic sensitivity test because of, unfortunately, Staphylococcus develop resistance to many antimicrobial medicines, including antibiotics and chemicals. Glucose installation. It is necessary to do any antibiotic sensitivity test, maybe this diffusion test, dilution test, ellipse test, and other. Because our doctor is waiting for results, doctor should the proper medicine for him in accordance with its results. Serological method used for chronic staphylococcal infections like osteomyelitis, septicemia, a modern test to detect antibodies against alpha toxins. Treatment. For antibodies are used according to the results of determining the sensitivity of the isolated cocci. Staphylococcal bacteriophage and polyvalent bacteriophages are used. A polyvalent bacteriophage includes other bacteriophages then also for treatment, antitoxic, anti-staphylococcal flora, or human immunoglobulin. This is the human leaves immunoglobulin that is used for treatment. And there's chronic infection, staphylococcal vaccine is used. Inactivated vaccine is like an exception for immune system stimulation. Specific prophylaxis for active immunization are used staphylococcal toxoid and neuro-associated vaccines. Staphylococcus pseudomonas aeruginosa. All these microbes are able to cause pathogenic infection. Staphylococcal toxoid is prepared of staphylococcal exotoxin, and it is in Latin anatoxin. Anatoxin staphylococcal. And I hope you remember production of anatoxins, toxoids. They are prepared of exotoxins. Those are treated by formaldehyde, and this mixture is incubated at thermostat temperature 38 degrees by Celsius during 3 or 4 weeks. And during this time, formaldehyde blocks toxic receptors of exotoxins, and finally we will get the same protein but without toxic protein. And this preparation is named anatoxin, and it is able to develop production of antibodies. Anatoxins or toxins are used for specific prophylaxis of toxin infection. And I gave this information on the convention. So, non-specific prevention. Compliance with the rules of asepsis, antiseptic, disinfection, and sterilization. Hospitals, of course, it's very important. And detection of bacterial carriers and their treatment, especially in hospitals and maternity hospitals. Carriers are very dangerous. Streptococcus, genus bacteria. Genus Streptococcus includes more than 20 species, and species of importance human physiology are two: Streptococcus pyogenes and Streptococcus pneumoniae. Morphology. Streptococcus pyogenes, small gram-positive spherical cells, arranged in pairs or short chains. There are no spores and flagella, during heavy capsule. They are located in chains. Streptococcus. Observation. Streptococci are facultative anaerobes. Blood supplemented media are prepared on solid nutrient media at a temperature of 35 to 7 degrees Celsius, and they form small grayish transparent colonies of oxide. According to the ability to cause hemolysis, they are divided on alpha hemolytic, beta hemolytic, and gamma hemolytic streptococci. Alpha hemolytic streptococci able to develop greenish color around the colonies. Beta hemolytic streptococci are able to develop clear zones around the and gamma hemolytic streptococci not visible hemolytic. Biochemical properties. Enzymatic activity is novel. There was staphylococci, and Streptococcus pyogenes breaks down some carbohydrates only glucose, maltose. Then, antigenic structure. According to the classification, Mansfield, scientist was Mansfield, Streptococci are divided into 20 serogroups named by letters from A to D. Serovariants are distinguished between the groups, and Streptococcus pyogenes belong to group A. Resistance. They die at temperature of 56 degrees by Celsius within 30 minutes. When boiled, they die instantly. Phytoreceptors. Capsule, cell wall complex, protein A, M, or fibril protein is able to bind immunoglobulins by Fc fragment of immunoglobulin neutralized by the protein. The complex is not right complex is not developed, so it's not recognized by macrophages. Aggression enzymes named streptolysine O, streptolysine S. These antigens are recognized by resistant antibodies. Pathogenic toxins are produced by strains of those are able to cause scarlet fever. Pyrrolidase enzyme for the production of phosphoric acid, streptokinase breaks down blood clots. It's a largest streptococci in the epidemiology. The source of infection is a sick person or a carrier. The main transmission routes are airborne, daily contact. Clinical manifestations. Acute streptococcal diseases in which streptococcus is the name of the only positive agent is scarlet fever. Scarlet fever is caused just by streptococci only. Erythema, tonsillitis, acute glomerulonephritis, endocarditis, sepsis, nephritis, kidney, and infections caused by streptococcus, scarlet fever, raspberry color tongue, and rash on the skin. This is clinical symptoms of scarlet. Then, follicular tonsillitis in response. Inflammation of the skin and the tissues under the skin. Then, chronic streptococcal diseases, rheumatism, rheumatic fever, and chronic tonsillitis. Chronic streptococcal infections. Immunity. Post-infectious immunity is unstable, unfortunately. Also, they differ by antigenic structure, so the immunity is not developed to all species. Microbiological diagnostics. Sepsis, osteomyelitis, and other types of generalized streptococcal. For others, they take fast discharge of mucous membranes spread with bacteria. Method of diagnostics. Cultural method, inoculation for the investigated material blood agar, followed by submission of full culture and identification. Identification means detection of species name. Then serological method. Serological method, detection of streptococcal antigen in the blood and in blood serum, detection of antibodies to streptococcus against antigens such as streptolysine or streptokinase. Important. Treatment. The main problem in the treatment of streptococcal infection is the resistance of the to antimicrobial drugs, the same like staphylococcus. Treatment is carried out in accordance with the results of the antibiotic sensitivity test. Streptococcal bacteriophage and polyvalent bacteriophage are also used. It consists in observing the rules of the sanitary and hygiene region, the rules of the asepsis, antiseptic, and sterilization in hospitals. Nowadays, molecular vaccines developed for specific prophylaxis of streptococcal infections. It's not included in national vaccination schedule, but it is developed. Molecular vaccine. Streptococcus pneumoniae. They are named in English pneumococci. Pneumococci are the main causative agents of bacterial pneumonia. Very often they are isolated from the patients with bacterial pneumonia. Morphology. Streptococcus pneumoniae are gram-positive diplococci located in pairs, usually along satellites, like lancets. They are looking like lancets. They form a polysaccharide capsule. They have no flagella, they don't develop spores. Pneumococci smear and pneumococci structure. So they belong to Streptococcus genus and they should be located in chain, but in human body and animal's body they are located in pairs, surrounded by macrocapsule. They have that lancet shape. They are located in it. And cultivation. They are facultative anaerobes. They don't grow on ordinary media. They need blood or serum. They grow on blood and serum media. On blood above small grayish polyps surrounded by a zone of hemolysis. they are able to produce alpha-human greenish color. Antigenic structure. There are three main antigens: cell wall polysaccharide antigen, then protein, and then capsule antigen. Based on the differences in the capsule antigen, pneumococcus divided into 20 pure variants. Receptors. Capsule. Pneumococcus without capsule are not able to develop pneumonia. So, capsule is very important factor. Substance C has enzymes, protease that is able to destroy immunoglobulins, uraminidase, structure uraminic acid in human being cells. Hyaluronidase help them to invade them. And pneumolysis, beta humanase. Epidemiology. Pneumococcus representatives of normal microbial flora of the upper respiratory tract are conditionally pathogenic streptococcus, and there are another species. Those are able to exist in the oral cavity, like Streptococcus salivarius, Streptococcus mitis, Streptococcus mutans. They are able to exist, but they are named conditional pathogenic because of, on the background of, if they have opportunity to multiply, they will multiply, able to cause the pneumonia and the genic infection. So the source of infection is a sick person and a carrier of bacteria by infected by Streptococcus pneumonia, and the mechanism of transmission of pneumococcal infection aerogenic, inhalation of droplets of saliva. Transmission route is airborne, indicated by droplets. The entrance gate is the mucous membrane of the orifice. Clinical manifestations. Pneumococcal infection manifests itself as a carriage or severe pneumonia, otitis media, bacteremia, and meningitis. Microbiological diagnostics. Material for research. Newton part of the cerebrospinal fluid. Methods are microscopical, cultural, biological read. Microscopical method. In preparation stained according to Brown staining, lanceolate diplopocci located in various defect. This is smear of sputum, so there are some particles of sputum on the other cells and the ground with diplopocci. Cultivation. The test material is inoculated onto blood agar. Then we are looking for analyzes biochemical and antigenic properties are studied for identification. Determined to sensitivity or bacteria to antibiotics by any antibiotic sensitivity test. Biological method is used because of pneumococci are able to develop colonies on nucleic acid, but sometimes there are not enough bacteria in the sputum, and sometimes they don't develop colonies, so mice are very sensitive to it. Okay, so the material should be injected, sputum should be injected into the mice, and followed by exhalation of pathogen, and then it is possible to inoculate lungs of these mice and blood on media and blood agar. It is possible to isolate pupils and to identify. So in this case, animals are used for culture isolation, for isolation of bacteria treatment. Many strains of pneumococci are resistant to penicillins. Drugs of choice: oral chemical, ceftriaxone, vancomycin, rifampicin. Clinical with antibiotic sensitivity test. Prophylaxis. For specific prophylaxis, polyvalent chemical vaccines containing capsule of polysaccharide antigens have been developed. Neomar 23, Previnar 13, Florix. So, not included in national vaccination schedule, but they developed molecular vaccines. Non-specific prophylaxis complies with the rules of asepsis, antiseptics, disinfections, and sterilization in hospitals. And again, identification of bacteria carriers and their treatment. Neisseria. Neisseria genus. Genus Neisseria includes many species, and some species are able to exist in oral cavity, like non-microbial. They are named non-pathogenic Neisseria. But two species are pathogenic for human beings, named Neisseria meningitidis and Neisseria gonorrhoeae. Neisseria meningitidis is named in English meningococcus. Neisseria gonorrhoeae are named in English monococcus. So Neisseria meningitidis, meningococcus. Morphology. Meningococcus are gram-negative diplococcus. In the form of conjugates, they are located close to each other by curved sides. They are located to each other by curved sides. They are located in pairs, and they have feeding, and they have microcapsule. They don't form spores and have no flagella. Oxidation and biochemical processes. Meningococcus are aerobal, which they need all two demanding nutrient media with blood agar serum agar. They are not able to grow for not the nutrient agar. They form small S-type forms in water, and they are not biochemically active, but they are able to ferment sugars. Antigenic structure. The antigenic structure is heterogeneous. Trace those belong to this species, they belong to one species, they serine indigenous, but strength differ by antigenic structure, and according to the capsular antigen, 13 serogroups are distinguished. So again, immunity is not developed to species, completely to the species. It depends on variant. Virus factor. They are gram-negative bacteria, and I hope you know that all gram-negative bacteria, they have lipopolysaccharide in the cell wall, and this lipopolysaccharide is a toxic substance for humans, and it is named endotoxin. And exactly meningococcus confess that coccus is very toxic and toxic, highly toxic. They don't produce any exotoxins, pay attention. They have just endotoxin. Meningococcus and gonorrhoeae are not able to produce any exotoxins. Compare with Staphylococcus and Streptococcus. Staphylococcus and Streptococcus are able to produce many exotoxins, and they are able to develop different clinical forms due to the specific exotoxin action. Neisseria have no any exotoxins. Memorize, it's very important. And they have just endotoxin. Anyway, meningococcal endotoxin is highly toxic. And the patient is able to die due to endotoxin. So they have endotoxin, lipopolysaccharide from the cell wall. So they have microcapsule. They have fimbrium for attachment, for adhesion. They are able to produce proteases for the functional hemoglobin. And they are able to produce two enzymes, isoamidase and uraminidase, that help them to invade. Resistance. They are vaguely resistant to external influence. They are adapted to human body temperature, and they quickly die under the influence of direct sunlight, temperatures, and temperatures below 22 degrees by Celsius. If meningococci are out of human body, and if temperature at room temperature, they will die. 22 is room temperature. I will die immediately. Cannot resistance absolutely. Epidemiology. Meningococcal diseases is widespread. In all countries, such infections are detected. In African countries, it is very often. The source of infection is the person, a sick person and a carrier of bacteria. Very dangerous carriers of meningococci. Cause of transmission route is airborne, and a sick person has high temperature, and a sick person stays at home or maybe admitted at hospital because of he has clinical symptoms, but carriers no clinical symptoms, and they have meningococci in oropharynx, and they have no any complaints, any clinical symptoms. They communicate with another people, and other people are infected by droplets. The meningococci are circulated just among human beings. I told you they are not able to survive out of human body, but not possible to eliminate meningococci from the earth because of carriers. And every carrier is able to infect other people. And even the carrier state is not stable. What does it mean, carrier state? The person was infected by pathogenic bacteria. And they are recognized by immune system and other defensive factors, but they are not able to eliminate completely bacteria. Bacteria are not able to multiply. A lot of to cause clinical symptoms. So it is a balance between defensive factors and bacteria, but it is not stable balance. And on any background of immunodeficiency, maybe cold temperature, maybe excess of sunlight again, uh, excess of sunlight suppress immune system. Sunlight, sun rays are not uh, harmful for immune system. They're not useful for human body. So on any background of immune deficiency, meningococci will multiply quickly, and like it was the person was a carrier yesterday, and today he is a patient. He has clinical symptoms, and the entrance gate and bleeding site of meningococci are most often the nasopharynx. So carriage is formed, which can develop into meningococcal nasopharyngitis. If meningococci will get opportunity to multiply, they will multiply and they will cause inflammation of the nasopharynx and nasopharyngitis. It is local infection, and of course, now it is a patient. This person is a patient, and treatment should be done. But unfortunately, sometimes clinical parents, doctors did not didn't pay attention to the sore throat, and without the treatment, meningococci are able to go inflammation of nasopharynx, and then they are able to go into the bloodstream, and they are able to multiply in the bloodstream. And it is, of course, the meningococci, which is sepsis for the meningococci, sepsis or septicemia, and exactly this clinical form is named meningococci. And due to LPS action, meningococci will develop high temperature, after still 40 degrees by Celsius. Patient has severe headache. But on this stage, such people are admitted at hospital immediately. But sometimes meningococci are able to go through the blood-brain barrier, and they are able to multiply in the brain, and they cause meningitis. And sometimes patients die of meningitis, not also just in the life. After the cancer diseases, a rather stable group specific immunity develops again. Meningococci differ by incidental structure, and the character of immunity is that they are just variant specific immunities developed. So after recovery, reinfections are very often. Microbiological diagnostics. Specimen mucus from the nasopharynx. If the person is a carrier or maybe has local infection nasopharyngitis, we have to take specimen from nasopharynx before transfer. In case of sepsis, blood should be granted, and in case of meningitis, cerebrospinal fluid should be taken. Spinal cord, it is traumatic procedure in the and microscopical method. If material is taken from the nasopharynx, microscopical method is not used. Because off, I told you that meningococci are able to exist in healthy people. Non-pathogenic species of meningococci are able to exist, so not doesn't make any sense to do microscopy. Detection of gram-negative diploid not indicates that they are exactly necessary meningitis because of not possible to identify microbes just by looking. So, but if the material or examination is blood or cerebrospinal fluid, the smears are stained just by methylene blue. Easy, it is easy because of ground staining method is complex staining method, and it takes time, so we have to get results quickly. So, and just this syringe is used, and this is smear, and you see there are many different cells, and there are tiny dots, the tiny dots, and they are located. They are able to multiply in macrophages. They are located out of cells, but they are not obligated intracellular cells. They don't need the host cell for multiplication. They are able to multiply out of cells, but if they are inside the macrophages, they are able to multiply inside. They are able to protect themselves from phagocytosis, and they are able to survive and even to multiply. Section of meningococi inside the cell has diagnostic meanings. Cultivation. Cultural message is used. The specimen is inoculated on blood agar, and then pure culture should be isolated via local colorless forms. And pure culture is identified by biochemical and antigenic properties. Serological message. Serological message is used. Detection of pneumococcal antigens in the cerebrospinal fluid by ELISA and the new first test. Then detection of antibodies in the blood serum is carried out using non-direct immunotest test. It is what to do with antibodies. For rapid diagnosis of course, antigens are detected in cerebrospinal fluid. It is for diagnosis. Then molecular genetic message is used now, and rapid diagnostic methods have been developed for the detection of specific parts of bacterial DNA by using polymerase chain reaction. Very specific and very the most sensitive, the most specific method nowadays. Treatment. Antibiotics of choice. Penicillins, third generation cephalosporins, cefotaxime, ceftriaxone, ciprofloxacin are used. Treatment and prophylaxis. No specific prophylaxis aims to identify and treat carriers of patients. Time by time, people should be children at schools, at kindergartens, students at hostels, soldiers and army should be exempt for detection of carriers of meningococi, and they should be treated before they will infect other people. They carry out active and passive prophylaxis among contact persons. For active immunization, clinical polysaccharide vaccines are used, consisting of antigens of meningococi, serogroups A and C. Do you see polysaccharide meningococcal A plus C vaccine? Groups A and J and P. Now it is improved and it contains polysaccharide groups. Just an example of Savinova vaccine. Passive prophylaxis is carried out by immunization with anti-meningococcal immunoglobulin or anti-meningococcal serum. Contact for people who was in contact with the patient. For emergency prevention, emergency prophylaxis, it is too late to vaccinate them. Then they need preparation both are used for people, like anti-meningococcal immunoglobulin or anti-meningococcal serum. Then Neisseria gonorrhea, pneumococci. They have the same morphology like the meningococci. They are negative diplopocci in the form of porphyrins, located in pairs close to each other by cells. And they have pili, coordination, and they have microcaps. They don't have spores and they have no flagella. The same like meningococci are. They have similar morphology. So not possible to recognize them just by gross staining. They are not negative. Cultivation of biochemical properties. Gonococci are aerobes. They are aerobic bacteria, and they are demand depleted. They are not able to grow on ordinary agar. They need serum agar or blood agar. The smallest type is on blood agar. They need blood agar. They are not biochemically active. They are able to ferment, but only So compare with Staphylococcus. Staphylococcus are the biochemically active protein, and they are resistant in nature. Cryptococci are less biochemically active, they are not so resistant in nature. And meningococci have low biochemical activity and two sugars only in gonopic one sugar only. And Neisseria are not resistant in nature, absolutely. Uh, Antigenic structure. The antigenic structure is complex. Strains don't belong to this species. They differ by antigens. Very complicated antigenic structure and contains not studied well. Anyway, it is conclusion was made that they are differ by antigenic structure because all reinfections are very common. After recovery, the person is able to be infected by a new strain of gonorrhea with another antigenic structure, and again clinical symptoms of gonorrhea are developed. Immunity is not developed to all strains of this species. Immunity is developed for 13 antigenic variants, so reinfections are very common. So they have somatic and capsule antigens, and the main antigens of gonorrhea are P, those for again. What are receptors of gonorrhea? Capsule and P, then cell wall surface proteins, and endotoxin and proteases. Again, pay attention. Gonorrhea, like meningococci, are not able to produce any exotoxin. They have just endotoxin and P and proteases. Resistance. Gonorrhea are unstable in the external environment. They die quickly when exposed to disinfectants. Immunology. The gonorrhea parasites only in the human body. The source of infection is a sick person, especially with a chronic or symptomatic form of gonorrhea and acute gonorrhea. The route of transmission is sexual, daily household. Infection of newborns is possible when passing through the infected first row of mother. Gonorrhea are not able to go through placenta. So the fetus is still sterile in mother's body, even though mother has gonorrhea. Fetus is not infected, but newborn will be infected due to direct contact with infected tissues of mother. And gonorrhea, what clinical manifestations? Gonococcal infection manifests itself in the form of virulent inflammation of the mucous membrane of the urinary tract, gonorrhea, and conjunctiva of the eyes, venom, and really gonorrhea has affinity to mucosa of urinary tract, and they cause affinity to mucus of eye. If they are on the eye, they cause venerea or gingivitis. Obviously, one that was infected from the mother. And microbiological diagnostics. Specimen pass from the urethra or vagina or cervix gingiva or pass. If they are pathogenic protein, the very pass will be developed. Microscope. The smear is stained with ground staining method and methylene blue staining. So again, they are able to survive in microphagia. This is smear stained by methylene blue, and it is possible to see cells located out of cells, and it is possible to see cells located inside the macrophages, so they are able to protect themselves from phagocytosis. And did you know that not possible to recognize, not possible to differentiate gonorrhea from meningococci? They are also similar in this way. Cultivation. For cultivation, the material should be inoculated on blood agar, and we are looking for colorless colonies. Then suspicious colony should be re-inoculated on the serum slab. Then we should isolate pure culture. And pure culture is identified by biochemical and genic properties. Serological method. Serological method is used in case of chronic gonorrhea monoclonal antigens are identified in the patient's material by the immunofluorescence test and for agglutination test. In chronic forms of the diseases, antibodies are detected in the blood serum because of in chronic forms it is difficult to isolate bacteria by cultural methods. Of course, molecular genetic method now is very widely used, and this mentioned the something molecular genetic laboratory and by the polymerase chain reaction is also to detect the specific part of nucleic acid. The aim of molecular genetic method is to detect specific part of nucleic acid of presumptive positive agent. So very often we are looking for DNA and if we're looking for RNA viruses, we are looking for specific part of RNA, viral RNA, very specific, the most specific and the most sensitive method molecular genetic. PCR is not the method name, PCR is just reaction, the phase chain reaction. And maybe many reactions are used, and maybe modified PCR is used, reverse PCR, PCR in real time, and the DNA zones and so on. Treatment. Treatment is, again, unfortunately, gonorrhea, these strains of gonorrhea differ by sensitivity to antibiotics. They did develop resistance to many preparations. So, just necessary to, again, to do antibiotic sensitivity test to detect sensitivity of isolated strain to medicines. So, those are useful treatment. Treatment is carried out according to the results of the antibiotic sensitivity test. And every patient with gonorrhea is treated by our own personal results. So, cephalosporins or fluoroquinolones are used in chronic gonorrhea. The killed vaccine is used for immunotherapy. It's an exception. I hope you realize that vaccines mainly are used for specific prophylaxis of infectious diseases, and absolutely healthy people should be vaccinated. For any case, maybe in future they will be infected by these microbes, maybe not. So we are talking about vaccines are used for prophylaxis, for specific prophylaxis. But there are some exceptions when vaccines are used for treatment of chronic infections. I told you about staphylococcus vaccine that is used for treatment of chronic infection caused by staphylococcus. And now there's this information about polio vaccine, inactivated vaccine, that is used for treatment of chronic gonorrhea. What is the aim of this procedure? Which means that the immune system of the patient, the person is infected by staphylococcus, maybe gonorrhea, the immune system is not able to develop response and to eliminate microbes from this patient's body because of some difficulties maybe in recognition. And if the pathogens are injected, the pathogens are injected in human body, they are recognized by immune system, and the immune system is activated, and it helps for immune system to develop immune response. And due to this immune response, of course, the treatment should be in complex, including medicines, including antibiotics, and this vaccine. And by the of many preparations, finally, it is possible to eliminate pathogens from human body. And then we will study herpes viral infection. In case of herpes viral infections, again, vaccine is used for treatment, not for prophylaxis. Again, the aim is to stimulate immune system, to help immune system to develop immune response. Prophylaxis. So, summarize, vaccine is produced, but it's not for prophylaxis. Vaccine is used for treatment of chronic gonorrhea if it is necessary. All over the world, there is no vaccine for prophylaxis of gonorrhea, also of strains of gonorrhea differ by antigenic structure, so not possible to produce vaccine. Next is not really, so no specific prophylaxis on gonorrhea, and just no specific prophylaxis, identification of the source of infection, and exclusion of casual sex, and use of personal protective equipment condoms, so safe sex is the main message of prophylaxis of gonorrhea. And for the prevention of gonorrhea in newborns, immediately after the birth, conversion, antibiotics is instilled into the eyes, and for the girls, addition of the mucosal membrane of genital tract, for prophylaxis of gonorrhea and gonorrhea for any kids. Only once we get this treatment. So the lecture is over. Have you any questions?
No, doctor, thank you.
Okay, so the next lecture, the last lecture, will be in 2 weeks from now, at the same on the Tuesday at the same time at 8 a.m. Now you have to answer questions on educational process just for the lecture, as usual. I will check attendance for the lectures in accordance with entering for test.
Okay,
Goodbye.
goodbye.
goodbye. أخذت الغياب يا جماعة.
هي بتاخد الغياب من الامتحان اللي بيتحل على البورتال.
نعم.
بتاخد الغياب